Hello, everyone. Today, we will introduce post-translational modification. There are 4 parts. Overview of post-translational modification. How does post-translational modification work? Most common post-translational modifications, impact on health and disease. Post-translational modifications, PTMs, are changes to proteins or peptides that are catalyzed by enzymes after completion of ribosomal translation, and it generally have a positive impact on peptide activity. PTMs alter the physicochemical properties of the peptide by altering the electrostatic charge, hydrophilicity, and conformation, thereby modulating their ability to specifically bind to the protein of interest. It plays an important role in virtually all biological processes, therefore, the synthesis of PTM peptides is crucial for many projects. Post-translational modifications are for functional needs, and in another case, the need to fold into a natural conformation. How does post-translational modification work? Most peptides are modified shortly after translation is completed to mediate proper folding or to direct the nascent protein to distinct cellular locations. Other modifications occur after folding and localization are completed to activate or inactivate catalytic activity. Proteins are also covalently linked to tags that target a protein for degradation. In conclusion, they are modified through a combination of post-translational cleavage and the addition of functional groups through a stepwise mechanism of protein maturation or activation. Types of post-translational modifications. Addition of chemical groups, hydroxylation, phosphorylation, acetylation, methylation. Amino acid modification, demidation, eliminylation. Cleavage, proteolation. Addition of complex molecules, ampylation, ADP ribosylation, glycosylation, prenylation. Addition of polypeptides, ubiquitylation, UBL protein conjugation. As we can see, sites that often undergo post-translational modification are those that have a functional group that can serve as a nucleophile in the reaction, the hydroxyl groups of serine, threonine, and tyrosine, the amine forms of lysine, arginine, and histidine, the diolate anion of cysteine, the carboxylates of aspartate and glutamate, and the N and C-termini. In addition, although the amide of asparagine is a weak nucleophile, it can serve as an attachment point for glycans. Rare modifications can occur at oxidized methionines and at some methylenes in side chains. Consequently, the analysis of proteins and their post-translational modifications is particularly important for the study of heart disease, cancer, neurodegenerative diseases, and diabetes. The characterization of PTMs, although challenging, provides invaluable insight into the cellular functions underlying etiological processes. Technically, the main challenges to studying post-translationally modified proteins are the development of specific detection and purification methods, including mass spectrometry, Eastern blotting, and Western blotting. Translational modifications can be used in many cases: cleavage and formation of disulfide bridges during the production of insulin. PTM of histones as regulation of transcription, RNA polymerase control by chromatin structure. Cleavage of polypeptide chains is crucial for lectin specificity. Especially, post-translational modifications of insulin are important for diabetes. At the top, the ribosome translates a mRNA sequence into a protein, insulin, and passes the protein through the endoplasmic reticulum, where it is cut, folded and held in shape by disulfide, SS, bonds. Then the protein passes through the Golgi apparatus, where it is packaged into a vesicle. In the vesicle, more parts are cut off, and it turns into mature insulin. Created peptides provides a variety of post-translational modified peptides for proteomics and epigenetics. In addition, we will make appropriate modifications to the peptide according to our customers' needs. Created Peptides is specialized in the peptide products and services, providing a confidential and efficient service at competitive prices. Every step of peptide synthesis is subject to Creative Peptides' stringent quality control.